Welcome to Lecture 2 of our Unit 7 Hereditary Genetics. We're going to start by talking about asexual reproduction. So, asexual reproduction is basically, it is exactly what the name says. It's reproduction without sex from one organism. It's important that you remember, though, that it does not lead to any genetic variation. Basically, the offsprings are identical. And many organisms are going to use mitosis to reproduce asexually through what is called budding. So here are some examples of asexual reproduction. We have binary fission, and basically it's using mitosis, and then we end up with the two identical daughter cells. We have fragmentation. You can see here with the planaria, and basically you cut the worm, it's in 3 different segments, it will regrow everything, and it now has clones. Over here we have what is called budding, and plants do this, and basically this little piece buds off until it falls off and grows into its own. Hydra does that. We have what are called runners. For instance, this wild strawberry, it basically develops these root systems, which are called runners, and then it supports those runners to get these new plants until their root system is developed. Also, another form of budding, yeast produces their clone basically through budding as well. So let's talk about the advantages and disadvantages of asexual reproduction and sexual reproduction. So first, we're going to talk about asexual reproduction. So some advantages. They are going to be able to survive better in a non-changing environment compared to sexually reproducing organisms because they can reproduce faster. Another advantage is that it's going to take less time because their offspring are basically simple and they don't need a lot of development. It's also going to take less energy for them to reproduce because energy is not used for behaviors and feature development necessary to find a mate. And on top of that, offspring usually do not need parental care. So some disadvantages is that there is no genetic variation, and they also do not survive well in changing environments. Okay, and before we go on, I love charts like this. However, I think that it would be better if we could, if you can just highlight somehow, which it's hard to do on the computer, but separate these two, okay? Asexual versus sexual there. Okay, so the advantages is sexual reproduction creates a population with genetic variation. They are also able to survive better in a changing environment due to their variation. So with that variation, a phenotype could result that allows for a more likely chance of survival. The disadvantages to sexual reproduction is that it takes more time and energy because you have to find a mate. Then you have to produce the structures related to the reproduction. And then you have to obviously wait for the offspring to be born. And then you have to raise the offspring. So let's talk about how variation can arise in asexual populations, because we know that it can. So we know that point mutations occur. We just talked about that. And they can occur due to radiation, chemicals, heat, as well as replication errors. So we've got some examples of that here. Obviously radiation, you know what that is. But we've got here, this is called conjugation. And then here in bacteria, basically this is the bacteriophages putting new DNA in there. And then we've got transformation over here because of the recombinant DNA that was there. So there's several different ways. Now, inheritance and epigenetic tags. You need to know this for the AP exam. We used to think that basically a new embryo's epigenome was completely erased and rebuilt from scratch. But what we know is that there is something called an epigenetic tag that remain in place as genetic information passes from generation to generation. And this is called epigenetic inheritance. And basically what this means is that a parent's experiences in the form of these tags can be passed down to future generations. And Lamarck's idea of acquired traits wasn't that much off base. So basically what I'm saying is that the diet that you choose, the environment that you live in, all of these things can affect your genes, basically, right? We've talked about that. But what we didn't know before is that those can be passed down. So we are going to talk about a Hereditary lab. And this, again, is something that the AP board, it's a recommended lab. We are not going to do this lab, of course. But basically, what's going to happen is there will be sexual reproduction in fungi as the result of crossing over. And then you're going to use the crossover percent to determine the location of a gene on the fungi chromosome. So, to understand the purpose of this lab, you have to understand a couple things. First thing is sexual reproduction in fungus and how both meiosis and mitosis are used to make reproductive structures that are called asci. And these asci contain spores. The second thing is linked genes. And these are basically genes that are on the same chromosome, and that's why they're called linked. So the lab setup is basically there's an agar plate, and it's going to be sown with two types of Sordaria. And it's going to have one that produces a tan spore and one that produces a dark spore. So the plus symbol means basically that this phenotype is called the wild type. So in this instance, it's the dark spore. TN over here means that the phenotype is due to a mutation. So basically, what's going to happen is that we put this stuff on the agar plate, and we're going to have this reproduction that occurs, and it's going to look like, little fans of fingers coming out. And this is asexual reproduction. And it's going to be in all of these. Okay, so that's going to occur. So these are asexual. However, where they grow and their hyphae meet and fuse, these lines here are actually sexual reproduction. Because the spores that form here, they are going to be a combination from two different parents. So if two hyphae from the tan parent fuse, then asexual reproduction is occurring. But since these are from two different parents, sexual reproduction is actually occurring. Okay, so part one is the sexual reproduction. So the fusing of the two hyphae cells from two different strains of fungi. The fusing of the nucleus, they form a diploid cell. There's replication of DNA. There's division by meiosis 1 and 2, and that is going to form 4 cells. And then division from meiosis is going to form 8 cells. And these cells are going to be haploid. So, this first division through the meiosis, basically it's going to go, let me get a different color here. It's going to go from 4, hold on, technical difficulties. 4N, and it's going to go, we'll have 2N, and then 4 of the N cells. Okay, versus here, we have 4 of the N cells, and we end up with 8 of the N cells. So let me see if I can draw that out a little bit so that you can understand here. So basically, we have a finger here from, we'll say, the tan. Okay? And then we have another one here, and it's from the dark. So I'm going to color that in. Oops. Dark. So basically, these two are going to combine. They're going to fuse. So then we end up with we're going to have half dark, half not. So we start out with N. This is now a 2N. And then we have DNA replication is going to occur. And then that is how we end up with the 4N. Okay. So it is important for you to understand that on the chromosomes there's a gene for spore color, and it's going to be tan or dark. So if we are looking here, basically what's happening is just what we just talked about. Okay, we have the finger here with the tan, and we have the one here with the dark. Okay, so these two have now come together, and we have fusion, and it has created this. So this here, we're going to have replication occur. If you notice, there is no crossing over. This here, we've got meiosis 1. Okay, so now we are at 2 in. And then here we have meiosis 2 happening. And now we are going to be each at N. So, we've gone from 1 nuclei to 4, right? This is what the line of the ASCII is going to look like. And you have to be able to identify the tan versus the dark. And it's kind of hard in this picture, but these here, these are the dark. So these 4 are dark. So there's 4 tan, there's 4 dark. So if no crossing over happens during the sexual reproduction, this is important, the spores will form a 4 and 4 pattern. So for instance, what we just looked at, 4 and 4. No crossing over happened. And you can see it here, 4 and 4 and vice versa. So if any other pattern of light or dark spores is found, then crossover did happen. It did occur. So here, no crossing over. We've got the 4 and 4. But these 3 all have different patterns, so crossing over did occur. So what you're going to be asked to do basically is determine the percent of ASCII showing crossover. So basically what I would do is pause, take a moment, go through and label the ones where you know that no crossing over occurred. So I'm just going to label here with a little dot. So there's 4 and 4, there's 4 and 4, there's 4 and 4. Oh, I missed one up here. There's another one. There's another one. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 10 with no crossover. And we have 13 with crossover. So to determine the percentage, we need to take that 13 divided by the total, which is 23. That's going to give us, we have to multiply that by 100%. That's going to give us 56.5%. So that is our answer. So why are we going to determine this? We're going to talk about that further on when we talk about the results of the lab. Okay, so part 2 of our linked genes. Linked genes that are found on the same chromosome. And we're going to measure the distance between these two in what are called map units. We can also measure the distance between a gene and the centromere in map units. So, let's do a practice question so you see what I'm talking about. How many map units is the UNC 75 from the centromere? So we're looking right here, and here's the centromere, and we've got map units labeled here, so we're not quite at 10. So the answer here would actually be 9 map units, or 9 MU. Another one, how many map units apart is the MAB20 gene and the UNC 75? So the MAB20 is at about at 11. And we know the UNC 75 is at 9. So 11 plus 9. We are at 20 map units apart. So crossover can happen between two genes, leading to chromosomes that are called recombinant chromosomes. And individuals that inherit the recombinant chromosome and show those recombinant phenotypes can also be called recombinants. So there are some rules that you need to memorize. If genes are farther apart from each other, or if the gene is farther from the centromere, crossing over is more likely to happen. If crossing over happens between two genes 5% of the time, then those two genes are going to be 5 map units apart. And this is something, both of these, just something that you have to know. Okay? So let's talk about why, if they're farther apart, crossing over is more likely to happen. So let's look here on the first one, gene A and gene B. If we were to break that chromosome, okay, there's only really one little spot that you could break it. Versus over here, if we look at gene A to gene C, well, we could break it there, we could break it there, we could break it there, we could break it there. So there's a lot more chance of crossing over happening. So let's do some practice questions. Okay, so looking at what we were just looking at, we know which two genes would show recombination more often, A and B or A and C. We know that it is A and C. So gene A and B cross over approximately 5% of the time. So how many map units apart are they? And we know that the answer would be 5, because remember, 5% is 5 map units. Gene A show recombination 22% of the time. Gene A and B, oh, I'm sorry, my apologies, that should be gene A and C, are how many map units apart? 22% of the time, that means 22 map units. And then the next one. Gene A shows crossover approximately 7% of the time. Gene A is how many map units from the centromere? Well, there's not a second gene, you will note. They're only talking about A. But we can determine that based on where A is, we can compare that to the centromere, so we know that it's 7 map units. Gene C shows recombination 29% of the time. So again, we know that it's 29 map units from the centromere. Okay, so here we're going to look right here on this diagram. If UNC 101, which is right here, and UNC 75 are 5 map units apart, we should see recombinant crossover or offspring 5% of the time. What percent of the offspring on G will be recombinant due to crossing over between the GLP4, so let's find that, GLP4, and the FOG1, and there's FOG1. So GLP4, it's about 20, 21, and this one here is at 4. So 21 plus 4, we're looking at 25% of the time. H. EGL30, which is right here, crosses over 14% during meiosis. What is the distance from the centromere to the EGL30? And that would be 14 map units. Okay, so those, honestly, they're pretty easy. They don't get much more difficult than that, but they can pull some other ones, so we're going to do some other examples here. So scientists can use crossing over frequency to estimate the distance between two genes, estimate the distance between a gene from the centromere, and map locations on a chromosome. So these are the ones that can get a little bit trickier. So genes A, B, and D are linked. The crossover frequencies for B and D is 5%, B and A is 30, and D and A is 25. Determine the relative locations of these 3 genes on a centromere. Or I'm sorry, on a chromosome. Okay, so I'm going to draw my very badly shaped chromosome. Hopefully yours is a little bit better here. So we know that B and D is 5%. So I'm just going to go ahead and put B here and D here. And this is 5. Okay? Because we know it's a very short distance. B and A is 30%. And D and A is 25%. So we could do A down here. And that could be our 30%. But then it says DNA is 25, and we know that this is 30. That would add another 5, so that would be 35. So that tells me that we need to put A down here, because that would be 25 between D and A plus the 5, so 30 between A and B. Okay? You want to determine the relative locations of 4 genes. Okay, so let's draw our chromosome here. So we're going to find A, B, C, and D and where they're at on the chromosome. You know that A crosses over with C 20% of the time, B crosses over with C 15% of the time, and A crosses over with D 10% of the time, and D crosses over with B 5%. Okay, so again, I'm going to put my B and my D, so I know that I've got my 5% here taken care of. So there's B, there's D. So now I've got to come up with A and C. So A crosses over with C 20%, B 15, and A 10? Well, we know that this is 5, so if we add another 5 here and make that A, then that's going to be 10% between A and D. And we know that A and C is 20% and B and C is 15. So if we put C down here, then that span would be 20 and the span between C and B would be 15. Okay? So these are honestly, they're guessing. It's just you putting a puzzle together is all it is. And it's not as difficult as it may seem. Okay, so we're going to talk about the results of that sorted area lab, now that we've gone over everything. So, first of all, we determined the percent of crossing over from this diagram, remember, as 56.5%. So, we're going to divide that percent by 2 to get the correct crossover percent. We're going to do this because only half of the spores in each ASCII are the result of crossing over. So our correct answer is going to be 28.25%. So with this information, we can conclude that the gene for spore color is located 28.25 map units from the centromere. Okay, so if they wanted you to put that on a diagram, then here's your centromere, say right here, 28.25, and then you would just label it. All right, that is all for this lecture. I will see you guys next time.